because it's about 20 after. So for those of you who are coming in, welcome to the second sub-session of the plant sciences session. We got a little bit ahead of ourselves, so we just had to wait for 5 minutes. And our next speaker and our last speaker is Amelia Ryan, and she's with the National Park Service. And she's going to be talking about managing for the recovery of a poorly understood endangered wetland grass.
Thank you. Okay, so I'm going to be presenting work that I did with Lorraine Parsons, also of the Seashore. She's the vegetation branch chief. So the species in question is Sonoma alopecaris, Alopecaris equalis, variety Sonomensis. It's a short-lived perennial rhizomatous grass that's found in marshes in Sonoma and Marin County. It's found in, it's been historically collected in Sonoma County from about 15 populations, and historically in Marin County it was collected from about 3 populations. It was initially classified as either Alopecaris equalis or as Alopecaris uvulatus, and in the 50s, after reviewing some specimens, they classified it as its own subspecies, Alopecaris equalis, variety Sonomensis. It differs primarily on length, anther color, and robustness of the plant. But of these historic populations, in the 80s and early 90s, as interest in the species was sort of picking up, they resurveyed as many of these historic populations as they could, and they found that it had been extirpated out of several of them, primarily because of a habitat conversion. And in fact, there was 3 populations in the 80s and early 90s where it was growing, but since then, it's been extirpated from 2 of those. Down in Marin County, it was known from 3 populations, one in Bear Valley, one down in Bolinas, and one on the peninsula proper. And when they resurveyed at that time period, they didn't find it growing in Bear Valley. They didn't find it growing in quite the same location they thought it was collected in the peninsula, but they found it in several other locations. And they also rediscovered the population down in Bolinas. Now, a lot of, interestingly, it's the type species Alopecaris equalis, variety equalis, from which it's differentiated, is actually a weed in Europe, much of Europe and Asia. It's a circumboreal species that's really widespread, especially weed and rice paddies. Now, at Point Reyes, a lot of circumstantial evidence suggests that grazing is actually very important to the success of this species. And going back to that to that population down in Bolinas, which was the first population rediscovered on Point Reyes, which we call Population 1. When they first discovered the species, it was in a grazed wetland. It was heavily impacted by cattle, and they were concerned for the species, so they fenced off the species from grazing. After they removed the grazing, this is what happened to the population. They reinstated grazing, they had a little bump up, but within a few years, it had completely gone extinct from the site. Here's another little area where grazing was removed, and this is what happened to that population, the species. Now, the species does fluctuate, and there can be other things that affect it, but... And then here's another species. This is on Abbas Lagoon. The species once occurred across this whole swale here. After the formation of the park, they wanted to put in a trail, and they fenced part of the trail off from grazing, or they fenced the trail off from grazing, and it cut through the swale. After they fenced it off from grazing, it disappeared out of the non-grazed section of the swale. In 2008, actually, they decided they got money to put in a boardwalk across the wetland, and we decided to take the opportunity to realign the fence and the trail back close to the trail and open up a little portion of the trail to grazing. And this is what happened the first 4 years after that was moved. We have, first year there's no plants, about 40. Or I should say, we don't actually measure plants, we measure inflorescences because it's a rhizomatous grass. We can't really distinguish individual plants readily. And then this is in 2011, and by 2012 we had about 4,000 inflorescences. And that's that drainage. Okay, this is the upper portion that's grazed, and this is the lower portion that's not grazed. You can see the distinct difference in vegetation of the two sites. Okay, that's all very well and good, but we would like to sort of test our hypotheses, make sure that there's not other factors going on. I should back up here. So grazing, the mechanism of grazing benefit is suspected to be the reduction of competition. There was one study of the species done in the past by Sasha Jennett, and she found weak effects of clipping around adult transplants. So for her simulation of grazing, she clipped around the plants. So she doesn't simulate any soil disturbance from grazing. But she suggested that reduction of competition early in the life cycle may be important.
You want to press escape?
Okay, let's see. Escape. This species, just to note, is also very palatable to cattle. They like to eat it. So even though they seem to increase in their presence, they also like to eat the inflorescences, so they may be reducing its reproductive output. So starting in the fall of 2012, the National Park Service and Fish and Wildlife Service funded a study in conjunction with, or in consultation with Hall Cushman of Sonoma State, to look at to sort of specifically quantify the effects of grazing on the species. And then also to evaluate the habitat requirements of the species in general, and then use this to plan new introductions and also to use it to plan better management. So for the grazing part, our hypothesis was that more Sonoma alopecuris will grow when cattle grazing is present than when it's excluded. So we did fenced grazing exclusions at 2 of our bigger populations, population 5, which I already showed you at Abbott's Lagoon, and population 3 down here at AT&T. What's interesting to note about these is that these have 2 different grazing regimes. Population 5 is subject to continuous grazing. Population 3 is subject to wintertime grazing, when the rancher that holds the adjacent lease lets them into that site for a brief period. We measured soil characteristics like nutrients, organic matter, moisture, soil texture, and mineral content, water level, plant community, and then of course we counted Sonoma alopecuris. So this is the grazed site. You can see the fenced exclusions, the ungrazed, and then here would be the grazed plot. You can see that the cattle have quite an effect on the soil. It doesn't look that pretty. There's a lot of open patches though, and a lot more sort of soil disturbance. And then this is population 3. This is the wintertime look of the site. And this is population 3 in the winter, and this is the grazed and ungrazed condition, much less difference. And so this is what that same population 3 looks like in the summer. So in year 1, we actually had no significant results. These bars are not, these 2 graphs show the seasonal grazing, the continuous grazing, and there was actually no significant difference. But we saw a sort of pattern that hinted at something where in the seasonal grazing, there was a hint that perhaps there was a more, at least average, plants in the grazed sites than the ungrazed. But in the continuous grazing one, we had more plants in the ungrazed area than the grazed area. At this point, the site had been closed off from grazing for about 5 months. Here and here. Another thing part of the reason why these were so large, the error bars were so large, is that we were working with perennial plants that were already established. So there was a lot of variation in the number of plants in each plot we had. So we're working with existing populations, and so at Medhard. Okay, so that's year one. Nothing significant, but a suggestion of a different response depending on the grazing regime. So in year two, we decided to add a seasonal grazing component to the Abbott's lagoon population where there's continuous grazing. So our hypothesis here was that more Sonoma albicaris will grow in sites where cattle are seasonally excluded than in sites with continuous grazing or with continuous exclusion. So I'm just going to present the results from the Abbott's lagoon population. Unfortunately, the drought kind of hit this population, this area pretty hard, and there's very little forage, so the cattle didn't really spend any time in here. There were essentially no results between the grazed and ungrazed. But this is the results from the Abbott's lagoon population. These are the inflorescence output in the ungrazed areas, the grazed area, and the seasonally grazed. I actually log transformed the data for analysis, and it was the seasonally grazed had a highly significant more output, reproductive output than the other plots, either of the other plots. And then even though it looks like there's essentially no difference between the grazed and ungrazed plots, if you compare their output from the year before the difference, there was no difference from the year before in the amount of output, the amount of inflorescence put out by the grazed plots, but the ungrazed plots had decreased significantly in the amount of inflorescence they were putting out. So in other words, they were very variable to begin with, but after a year, these ungrazed plots had lost a lot of reproductive output or inflorescence output. So our hypotheses, more albicaris will grow when cattle grazing is present than when excluded. Yes, if you look at it at the change between years. And more albicaris will grow in sites where cattle are seasonally excluded than sites with continuous grazing or continuous exclusion. Yes. So using the results of the study, we actually applied for, essentially right away, and received a U.S. Fish and Wildlife Service grant to implement a permanent fence around population 5 to start for seasonal grazing exclusion. The cattle will be allowed in most of the year, but excluded during inflorescence production. This is expected to be in place this growing season. This is what it's going to look like. We have a fenced area with gates that will be open most of the year and closed in the summer. We're also using the data we collected as a part of the study to plan several new introductions. There have been some previous introduction efforts, but unfortunately they all seem to have failed. Meta-analysis of 25 rare wetland plant species and 15 introductions suggests that similarity to the source population habitat and introduction site was the greatest predictor of success. So we decided to focus on site similarity. We surveyed 12 swales. We actually discovered a new population in one of them while we were surveying, so job done on that part. We collected data on plant community and soil as we had done at the existing populations. And we also collected it from from our new population we discovered and from another large population. And then we used a technique called non-metric multidimensional scaling to compare the plant community and soils of the potential introduction sites to the existing populations. Unfortunately, we found that our two existing sites were extremely different from each other. They were the most different of any of the sites that we sampled as potential introduction sites. They're different, basically polar opposites in the amount of sand versus clay, calcium to magnesium ratio. This one was associated with high nutrients and organic matter. So very little clear environmental signal here. But what is interesting is those two other populations where it occurs, we also sampled, and this population 14 fell pretty close right in with the Abbott's lagoon population, and this 8.3 fell in pretty close with AT&T. So this suggests that possibly there's not just random locations where it occurs, but perhaps a few different models of sort of habitat the species can occur in. And so we picked for our potential reintroduction sites this population, which was most similar to the AT&T sites, and these two populations, which were most similar to the Abbott's Lagoon site.
Just do a
Okay. And so these are the three sites we chose. These two sites are similar to the Abbott's Lagoon population. This site is similar to the AT&T population. And so our introduction plan as it stands is to grow individuals from seed collected at both sites. We will, and these are actually baby Allochirus plants, and then take the plants and actually plant them in experimental plantings at both sites of each source at each site. Because though we suspect there's similarities, that there may be, some sites are more similar to their host sites and this may benefit the plants. We don't really know that. We're making assumptions that we've captured the data, that we've captured the features that are going to lead to reproductive success for the species. So we're going to plant individuals grown from both populations at each site and evaluate whether more similar donor populations are better adapted. If yes, it may indicate, first of all, ecotypic adaptation in the existing populations, such that the new more similar populations, in other words, if more similar soil types and plant communities indicate a better success, it may indicate ecotypic adaptation and difference between the two source populations. And secondly, we'll find out whether this leads to better success. More similar source populations leads to better success in the field. And of course, we want to do more after that. But the point is, we're hoping to use studies to better target our management to allow us to make better management decisions. For example, how to manage grazing and where to place new introductions. But we're also trying to incorporate studies into our management actions so that we can better assess them and gather data for future, such as, does the source population affect introduction success? What does that mean about that, those source populations? And I have lots of people to thank. Naturally, I have an army of seasonals and interns who did a lot of the work here, and several staff from Point Reyes, and Valerie Bloom in particular from the U.S. Fish and Wildlife Service has been really helpful, and Hall Cushman from Eastern State. And of course, my co-author, Lorraine Parsons. That's it.
you factor in the rainfall.
What do you mean? Like differences between years? Well, in what sense?
Well, that they might grow better if they have more rain.
In general?
Yeah.
Oh, you mean yes, we have definitely looked at rainfall, but in general, and we've done some modeling of trying to look at what habitat or what environmental factors respond to year-to-year variation, and it isn't clear that it's rainfall per se, but there definitely are patterns across populations where they seem to vary on certain years. A lot of them do better, but not all.
On your population 14, I would assume that that's has a long history of seasonal grazing.
The population 14, the newly discovered population. Oh, sorry. Okay, sorry. So population 14, he was asking about, that's the newly discovered population, and he's saying he assumes there's a long history of seasonal grazing. Yes and no. There's a long history of grazing there. The grazing is, the ranchers determine the grazing regime that they do. They do remove the cattle seasonally from the site, but there's no specific seasonal grazing regime put on it. So it's sort of intermediate level of grazing. But not necessarily a specific seasonal grazing. Some years it might be a lot longer and other years shorter. Yes?
Hi, I was wondering, during your study on the grazing, did anyone consider the effects of cattle trampling and dispersal of rhizome segments seasonally at a time of year when they'd actually be dispersed, have time to grow out, and with cattle being, for instance, with winter-only grazing, they would have a chance to grow and produce more culms as a result of that dispersal of rhizome segments during the wet season. Did anyone look at that?
No, that's actually a good thought. And in fact, I found another population a few years ago too, and I really suspect that it was that is exactly what happened. That it was dispersed by cattle, either by rhizome fragment or by seed caught up in muck, essentially on the hoof, because it was right adjacent in a wet area, right adjacent to a cattle trail. It's actually like a really kind of not beautiful site, just like a mucky edge of a cattle trail that I found right now.
Yeah, just as an afterthought, because that's one mechanism that I've seen duplicated with some of the invasive plant species like Pennymore and wetlands.
Mm-hmm. Interesting thought. Yeah.
The populations that failed from the earlier efforts to restore them in 87 and whatever, 2002, do you know why those failed?
No, because I was not here, and they initially established, and then they kind of went down over the years. And in some instances, it seems like not appropriate habitat was chosen. But in other instances, it's kind of actually hard to say, but why they failed per se.
Were they graze sites?
They were all graze sites. Well, actually, no. The 87 one, there was one that was on a non-graze site. They tried to reintroduce in Bear Valley and was like a spot where they were disturbing anyhow, and that didn't work at all. But other than that, they were all graze sites. But they varied in their, some were in ponds, which maybe where naturally generally occurs at Point Reyes, at least in swales, but some are in ponded areas within the swale. So, however, in Sonoma County, it occurs at Ledgeston Marsh, which is like a big kind of